In this session, we can discuss about chromosomes. This is the diagram of a cell, and you can see the nucleus has round bodies, and within this nucleus there are membrane bound structures which will undergo, these are the membrane bound structures that will undergo coiling to form shortened structures like chromosomes. Chromatin reticulum is undergoing coiling to form the chromosomes. So we'll learn in detail what is the structure, origin and different types of chromosome in eukaryotic cells. So chromosomes are the first carriers of genes, and hence they control metabolism of a cell. They carry the genes to next generation, and hence they are called vehicles of heredity. First, these structures were observed by Hofmeisters, and the name chromosome was given by Walder. These are the bodies which are found colored in nucleus during staining, hence they got the name chromosome, colored bodies. Within a species, number of chromosomes are common, but among the species, number of chromosomes will vary. In the cells of a species, the chromosome will be a constant. Number of chromosomes will be a constant. In the case of prokaryotes, chromosome is a genophore. It's a single DNA molecule. Eukaryotic chromosomes are formed of four kinds of molecules. They are DNA, RNA, basic proteins and non-basic proteins. Depending on the function they are performing, or the chromosomes are classified into autosomes and allosomes. Autosomes are the chromosomes which do somatic functions, and the allosomes do the heredity related things. A pair of similar chromosomes seen within a cell are called homologous chromosomes. Coming to the morphology of chromosome, they have a length of 0.1 to 30 microns and a diameter of 0.2 to 2 microns, and often chromosomes of plants are larger than animals. Chromosomes are visible only during cell division. Other time they will be distributed as chromatin reticulum within the nucleoplasm of the nucleus. A mitotic chromosome has two arms, a centromere, a primary constriction and telomeres. If you see the diagram of a chromosome, it has a constriction on the middle, and that constriction is called primary constriction or centromere, and it will divide the chromosome into two on a mid position, and the two sides, two parts formed by the constriction or by this primary constriction are called arms. The end of chromosome is called telomeres. If you see the morphology of chromosome, a chromosome is having a length of 0.1 to 30 microns. They are having a diameter of 0.2 to 2 microns, and they are observed during cell division. At other times they are seen in a cell as chromatin reticulum within the nucleus. It has two arms, and these two arms are formed by a constriction present, and this is called a primary constriction, and or otherwise it is called centromere. The presence of centromere divides chromosome into two parts called arms. The end of the chromosome is called the telomeres. Telomeres are stable and non-sticky ends, and they are the non-functional heterochromatic region. They are formed with heterochromatic regions which are non-functional and mainly formed of repetitive DNA. The sequences of telomerase in all chromosomes of a species are similar. Telomeres prevents end-to-end fusion of chromosomes, and they protect chromosomes from nuclease activity. During chromosome replication, telomerase are having significant role. Telomeres possess an enzyme called telomerase. The enzyme telomerase is concerned with aging of an organism. Cell division has 4 stages: prophase, metaphase, anaphase and telophase. During interphase, chromosomes are disorganized and they are seen in a cell as chromatin reticulum. During metaphase, a chromosome is linearly divided into 2 equal parts, and this linear half of a chromosome is called a chromatid, and thus a metaphase chromosome contains 2 chromatids. And these chromatids before metaphase are found to be elongated and loosely arranged, which are very long, uncoiled structures. They are called chromonimata. The chromonimata, when it condenses, chromatids are formed. Small granular bead-like structures present on chromonimata are called chromomeres. They are called chromomeres. The bead-like structures present in chromonimata are called chromomeres. The chromosome is having an outer covering, and that is called a pellicle, formed of non-genetic materials. Let's see the details of centromeres. Centromere is a clear area within the primary constriction of a chromosome. A chromosome is having a primary constriction. Within this primary constriction, there is a clear zone is present, which is called centromere. It is formed of constitutive heterochromatin with highly repetitive DNA sequences. Constitutive heterochromatins are heterochromatins which remains in a highly coiled condition throughout the life of that organism. Centromere also possesses a bundle of microtubules and a thin disc of granule called kinetochore. Kinetochore is otherwise called spindle spherule. The repetitive DNA present in chromosomes are also called satellite DNA. These are small repeats. Satellite DNA are small repeats of DNA present in the chromatin reticulum. The kinetochore contains a protein called kinetochore protein, or they are called as molecular motors. They help the movement of chromosomes through spindle apparatus towards cell pole during anaphase movement of chromosomes. The spindle apparatus attached to chromosome during cell division at kinetochore. And if a chromosome is having single centromere, it is called monocentric. When they are having more than one, they are called polycentric chromosomes. And if they are having no centromere, they are called holocentric chromosomes. Apart from primary constriction, these chromosomes have another second constriction, that is called secondary constriction. Apart from primary constriction, chromosomes also have another constriction, and that is called secondary constriction. The secondary constriction will be located towards end of an arm. And the secondary constriction are of two types, type 1 and type 2. The secondary constriction type 1 contains genes which code for 18S and 28S rRNA, and these RNAs are present in nucleolus and they are concerned with organization of nucleolus. Secondary constriction 2 represents the site of fission and fusion of chromosome segments, and their function is synthesis of RNA. So a chromosome contains centromere, arms, telomere and secondary constriction. Due to the presence of secondary constriction on chromosome, it will make a knob-like region on the tip of chromosomes beyond secondary constriction. This region is called satellite or traband. and the satellite bearing chromosomes are called SAT chromosomes, and SAT chromosomes region contain a compound called sinuacid thymonucleinico. That is the full form of SAT. The SAT chromosomes, when they are able to form nucleolus, they are called nucleolar SAT chromosomes. A chromatin reticulum or the disorganized chromosomes contain two kinds of structures depending on their coiling. The chromatin reticulum existing in highly condensed method are called heterochromatin, and the chromatin reticulum which are loosely arranged are called euchromatin. This is the heterochromatin which are highly coiled, and this is the euchromatin. Some regions of heterochromatins are permanently condensed throughout the life cycle of an organism. They are called constitutive heterochromatin or permanently inactive heterochromatins. But some regions of heterochromatin will become active during some stages of life cycle. Actually, these are euchromatic regions which will undergo condensation and coiling to stop gene action. These regions of heterochromatin which are temporarily heterochromatized or coiled are called facultative heterochromatin. Facultative heterochromatization is a part of gene modulation. Mainly depending on the position of centromere, chromosomes are divided into four types. They are telocentric, metacentric, acrocentric and submetacentric. In telocentric chromosomes, the centromere is occupying. In telocentric chromosomes, centromere is occupying a tip position. So it will be having only one arm. Such kind of chromosomes are called telocentric chromosomes. In acrocentric chromosomes, the location of centromere is subterminal, just closer to one end of the arm. One arm of the chromosome will be too large and other will be too small. One arm of the chromosome will be too large and other will be too small. This kind of a chromosome is called acrocentric chromosome. And the third group is called a metacentric chromosome, where the position of centromere is on the exact middle of chromosome and it divides chromosomes into two equal halves. This is called a metacentric chromosome. And the fourth group is called a submetacentric, where the centromere is moved towards one side of the arm, or the position of centromere is slightly away from the middle position, and they are called submetacentric chromosomes. So these are the four categories of chromosome depending on the position of the centromere. If you see the chemical composition of chromosome or chromatin, materials or the chemical components or the molecules which form chromosomes are togetherly called chromatin. The chromatin is formed of 95% deoxyribonucleoprotein, DNA and protein, and the remaining 5% is called a residual chromatin. Deoxynucleoprotein is formed of equal amount of DNA and histone, the nuclear proteins. Histones are the nuclear proteins. The quantity of chromosomal DNA is a constant in somatic cells. DNA content of cells of a species are constant, and this constant value is called C value. The 5% residual chromatin is formed of RNA, non-histone protein and metallic ions. The nucleoproteins present in DNA are basic proteins. The proteins present in chromosome are called nucleoproteins. Nucleoproteins are of two types, histones and non-histone proteins. Histones accounts for 80% of nucleoproteins. They are absent in prokaryotes. Histones have high molecular weight, they are water soluble and basic with arginine and lysine amino acids. They do not have the tryptophan amino acid. The proteins, histone proteins, are complexed with DNA through electrostatic bonds or ionic bonds. Histones are highly conserved and they are categorized into 5 major classes: H1, H2A, H2B, H3 and H4. The major roles of histones in genetic mechanism are: they control transcription of messenger RNA, they serve as gene repressors, they do folding and packing of chromosomal DNA into small units called nucleosomes. Coming to non-histone proteins, non-histone chromosomal proteins are called NHCs. The non-histone proteins are otherwise called chromosomal proteins or NHCs. Most of them are acidic and few are neutral. They include enzymes, structural molecules and gene regulators. Protamines are a group of non-histonic proteins and they form complex with chromosomal RNA and helps binding with histones. Non-histone proteins form complex with chromosomal RNA and thus they help the binding of RNA with DNA. They mediate and control DNA replication and genetic transcription. They stimulate gene action and serves as positive regulator in gene expression. Lastly, how chromatin or DNA is organized within a chromosome. If you see this diagram, DNA initially undergo a coiling with histone molecule, thereby forming a structure called a nucleosome, and then this nucleosome will be repacked within a chromosome. So let's see how this nucleosome model is. Let's see what are the features of this nucleosome model. The molecular organization of chromatin is best explained by nucleosome model, or it is otherwise called a solenoid model, proposed by Roger Kornberg and Thomas, and also later by Finch and Klum. According to this, chromosome has a fibrillar organization. It is like a beaded string with a series of regularly spaced bumps and noops. The beaded units within chromatin fibers are called nucleosome or nu bodies, which are the structural units of chromatin. A nucleosome unit is formed of 200 or more nucleotides, all class of histones and non-histone chromosomal proteins. A nucleosome unit is formed of 200 or more DNA nucleotides, all classes of histones and non-histone chromosomal proteins. If you see a nucleosome, these nucleosomes are having a core region and a linker region, or a core particle and linker particle is present. A nucleosome core is formed of 8 histone molecules compactly arranged to form a histone octamer and 1.75 turn of DNA coils around histone octamer, and now it becomes a core nucleosome. The histone molecules present in this core are H2A, H2B, H3 and H4. The DNA which turns around the core contain 146 base pairs. Adjacent to this core region, you can find small joining structures called linkers. Linkers connect adjacent core particles together. Linker region consists of a small stretch of DNA coiled around a histone molecule, that is H1, and the DNA of a linker region is 54 base pairs. A core particle and a linker togetherly called chromatosome. Folding of a nucleosome chain to a solenoid structure result in the extreme condensation of the chromosome. So chromosomes are the genes. So what we discussed today, chromosomes are the hereditary carriers. They are made up of DNA, RNA and proteins. The DNA will account for 95%. The remaining 5% are formed of RNA and other proteins. Chromosomes are having an external covering called pellicles. They are having two arms. Arms are formed by a constriction called a primary constriction. The tip of the arms are called telomere. Telomere contain an enzyme called telomerase. Apart from primary constriction, they will be having another constriction. This is called a secondary constriction. The secondary constriction will make a projection on the tip called a satellite, and the chromosomes which are having the satellite regions are called satellite. The satellite region contains highly repetitive DNA. They are otherwise called satellite DNA. Depending on the position of the centromere, chromosomes can be telocentric, acrocentric, metacentric or submetacentric. Metaphase chromosome contain two equal halves called chromatids, and the chromatids in a loosened form is called chromonymata. In chromonymata, highly coiled granular structures are present which are called chromomeres. The proteins present in DNA include both histones and non-histones. The most accepted model of chromosome formation is solenoid or nucleosome model, where histone molecules other than H1, 8 molecules join together to form a group. The DNA will turn around, that will form a nucleosome or a core particle in a nucleosome. The core particles are connected with linkers. Linkers are formed of DNA and H1 proteins. The chromatin reticulum is formed of these nucleosomes. This concept is called a nucleosome model of DNA organization, and it will help extreme condensation of the chromosomes or chromatin reticulum. And thanks for your patient listening.